We acknowledge that we are on Treaty 6 territory, the gathering grounds of many diverse First Nations, Métis, and Inuit peoples whose footsteps have marked this land and whose presence continues to enrich our vibrant community. Hello and welcome back to Research Recasted, the Knowledge Mobilization Podcast. I'm your host, Kelsey Johnston, and today we are joined by Dr. Ben Bourrie. Dr. Bourrie is a microbial ecologist with a particular interest in food fermentation. His research investigates the role of microbial interactions in shaping traditional fermented foods and their qualities. Thank you so much for joining us today, Ben.
Yeah, no problem.
Yeah. So to start, I was hoping if you could just kind of briefly give us an overview of the research that you're working on.
Yeah, of course. So I am a microbial ecologist, which means that I'm interested in how communities of microbes interact and behave and how things like the environment they're in or interactions between specific microbes influence those behaviors and outcomes, maybe. Specifically, my research focuses on microbial ecology in fermented foods. So I tend to look at how different fermentation conditions, so temperatures or oxygen, things like that, or community compositions and specific interactions between the bacteria and yeasts in these foods influence outcomes in these foods. So for me, outcomes are things like potentially like a health benefit in something that's like a functional food or a probiotic, if you want to use that term, or things like flavor and consistency of the finished product. So those tend to be the two main things that I'm focused on currently.
Okay. And just to clarify, by community, you mean like a bunch of different ones that kind of come together?
Yeah, exactly. So groups of different bacteria, sometimes different yeasts, different fungi. If you've ever looked at like blue cheese, so the blue streaks in the blue cheese are actually, that's mold that grows in the cheese. It's penicillium mold and it's blue, and that's where the blue color comes from. So that mold will work together with bacteria that get put into the milk when they first make the cheese. And those communities of microbes interact with each other to create the very complex finished product that you get when you have like a Rochefort cheese or something like that. Yeah.
Okay, interesting. Yeah, just the term community. I think that's a cool way to look at it.
Yeah.
So what inspired this research for you? Why did you choose to specifically study microbial ecology? Microbial
food specifically.
Yeah, fermented foods.
So I did my undergraduate in medical microbiology and immunology, actually.
Okay.
And I was really, in that time, particularly interested in bacterial pathogenesis, which is how bacteria cause disease and how they interact with the host environment. My undergraduate thesis was in how cystic fibrosis pathogens react to low oxygen environments that occur in the cystic fibrosis lung because there's buildup of mucus. And so I've always been interested in, how things like environment and host interactions, so human immune system with the bacteria influence microbial behavior. But I've also just always liked food and beer and things. In my undergraduate, I homebrewed beer with my brother-in-law, and we would make beer at home. And during my fourth year, I needed a class to fill out my full-time student status, basically. And I found out that the food science program at the U of A, where I was doing my undergraduate, had a class on brewing, enology, which is winemaking, and fermented foods at the 400 level. So I just took that class because it sounded fun. And then throughout the class, I kind of really liked it. It scratched a lot of those itches I had, where it was oh, when we have these microbes together, the food comes out different, or when you change this about the fermentation, this can affect the flavor. And as someone that liked both microbes and foods like that, it really just kind of sparked something where I was I can take what I really like about microbial interactions and microbial behavior and what I like about food and kind of pair them. And so at the end of the semester, because this was the last semester of my undergraduate program, I was planning on going to graduate school but wasn't 100% sure. And I just went to the professor that taught that class unannounced, went to her office and asked if she knew of anybody in the department that was looking for graduate students. And she just walked me down the hall to this other professor that had just gotten funding for a project looking at kefir, so fermented dairy product, and potential health benefits associated with that product. So it kind of, like I said, it combined my interests in microbes and how they behave and interact with their environment and the foods. And then I also got the added benefit of like health benefits and helping people, because that was part of why I also got into it. I'm the classic, like, first year in my biology program, I was going to med school, and that like clearly wasn't going to work. So I had to rejig, and that's totally fine because I'm much happier doing this, actually, I think. So, yeah.
No, that's so cool. I like how you kind of went from looking at diseases and all of that to more into like the health benefits, which is what I want to get into next, because you are working on a project with kefir and the potential health benefits of that. So maybe just start, what exactly is kefir?
So kefir is a fermented dairy product. It's almost like a drinkable yogurt in terms of its texture, but it is fermented instead of just bacteria. Like yogurt is typically just fermented by bacteria. Kefir is fermented by a mixture of bacteria, lactic acid bacteria. So most people will have heard of like lactobacillus or lactobacilli. So groups of them, and a large yeast population too. So it's a little bit different in that sense. It's also different in that traditionally it's made with what's called a kefir grain. Most people are not familiar with kefir grains, but they are maybe more familiar with the idea of a kombucha SCOBY. So a kombucha SCOBY is like this kind of slimy disc that you put in the kombucha to ferment it. A kefir grain is a similar idea in that it is a symbiotic community of bacteria and yeast, which is what SCOBY stands for. And it is a mixture of what we call exopolysaccharides. They're just long carbohydrates that the bacteria secrete into the environment. And it forms this little grain that looks almost like a cauliflower like floret, but like very small. It's like this big at max. You put that into the milk, the organisms that are in there start to consume the sugars in the milk, and they make things like lactic acid and esters, which can provide flavor. It could be slightly carbonated even. And so this is how you would traditionally make kefir. It's been around for over 2,000 years, and it is just like more common in Eastern Europe, but it's more recently been getting popular over here in the Western world as well.
So I know we had mentioned that there are health benefits to kefir. What are those health benefits?
So the health benefits that I was interested in my PhD and continue to be interested in are things related to metabolic diseases, specifically things like hyperlipidemia, so high cholesterol, LDL cholesterol, like bad cholesterol, being able to maybe lower that, increase HDL or good cholesterol. Things related to cardiovascular disease, which can be cholesterol, but also some other factors that we look at, fatty liver disease, that kind of stuff. There are studies out there in animals that show potential immunomodulatory benefits, so things like decreasing inflammation, potentially helping with allergies. There's studies that purport anti-cancer, but most of these studies are just in animals, and I'm always hesitant to kind of like be too gung-ho about health benefits, because a lot of the times things don't translate from animal to human studies. And that's what we're doing right now. Is during my PhD, I looked at the ability of kefir to improve things, like I said, like fatty liver disease, high cholesterol levels in mouse models of obesity. And what we found was that these traditional grain fermented kefirs did a better job than what was commercially available in the stores. And that's because they use different microbes. So most commercially available kefirs, not all of them, they're getting better, use different bacteria and yeast species and strains than what is in a traditional like grain fermented kefir. And so that's kind of what we found at the very beginning of my PhD when I was first doing this, and we've been moving on from that since then. After we found that the traditional microbes or traditional kefir did a better job, what I did was we worked on isolating those microbes, because kefir grains are great if you're fermenting kefir at home, but a lot of people don't have the time or desire to make their own fermented foods at home. And it's not really feasible to use a kefir grain to make 200,000 liters of kefir. If you want to make an industrial production of it, you would need hundreds of pounds of kefir grains and somewhere to store them, and you have to keep them fresh and upkeep. So the goal throughout the PhD was to isolate organisms out of the traditional kefir. So that involves things like making kefir, and then we use microbiological like media, so little petri dishes that you grow, and then you can isolate the individual organisms that are present, but you don't need that kefir grain. We isolate all these organisms, and then we can put them back together in what we call like a reconstituted community. So the benefit of that is that we have complete control over who's there and who's not, basically.
Okay.
Yeah. So I can just keep going if you want.
Yeah, I mean, This is so interesting.
Yeah. So from that, in my PhD, again, what we did was try different combinations of bacteria and yeast, starting with the ones that are most commonly present in the kefir grains and traditional milk fermentations themselves, and specifically used ones from our traditional kefir that did the best job at improving these like lipid related outcomes. And we eventually were able to find a combination of these organisms that, when taken individually and put back together into milk to do a fermentation, like you would on like an industrial production scale as like a starter culture per se, they were able to recapitulate the health benefits. So they had the exact same outcomes in the mouse trial as the traditional kefir did, in terms of they were able to lower plasma cholesterol levels, mostly LDL cholesterol. They were able to improve liver lipid deposition, so fatty liver disease related things, and a couple other markers related to cardiovascular disease as well. And that was kind of where I got to in my PhD. And since then, I've continued working on a postdoc with like Ben Willing at the University of Alberta, who was my supervisor and is now my collaborator on this. And we, in 2023, published a small human trial that showed that these benefits actually carry over into humans. So we were able to show that this specific mixture of microbes lowers LDL cholesterol and improves other cardiovascular disease risk factors in a group of human participants that had moderately high cholesterol levels. And so that's really encouraging, because a lot of the time these results, like I said, don't transfer from animals to humans. So
wow, that's so cool. Yeah. Actually being able to just like isolate the specific things so that you can kind of, I guess, replicate it consistently.
Yeah, the idea is traditional kefir, like I said, is it works. You can't really, some places will use it for industrial production, but really large scale industrial production, it's not that feasible.
Yeah.
But there's dozens of organisms in there, and we don't know what is responsible. So by isolating the organisms out, like I said, you can then add and subtract and tweak and change things exactly how you want, which you can't do with a naturally occurring community. You can't just go in and like pluck out the microbes of a specific type from a kefir grain. It's not possible. So this lets us toy with and tweak the community. And we've done things like, when we pull certain groups out of this kefir fermentation, the health benefits go away. So we know that like certain groups of organisms are important, and we do think that they actually are working together, because when we remove either the lactobacilli, so those lactic acid bacteria, or the yeast from the fermentation, the health benefits go away when either one of them is removed individually. So we think that they actually work together to produce some sort of metabolite, so a byproduct of the fermentation that actually provides these health benefits when it's consumed.
Oh, wow.
Yeah.
Wow. And just out of like my own personal curiosity, because I don't have a lot of experience with kefir, is it something that you can get, like you said it has like a yogurt consistency. Do you like just consume it on its own, or is it like part of different things?
So when we conduct our human trials, we give participants like a certain amount. It's usually about a cup serving per day. Some of them will put it into a smoothie. Some people will actually pour it on cereal. It is like a little bit acidic and like almost like fruity, yeasty like fermentation. So some people like that. You can also just drink it as is. some people will put like Nesquik, like chocolate syrup in it and drink it like that and just make it like chocolate milk basically. So you can kind of consume it however you want. Like you could replace milk with it basically, right? If you normally make like a fruit smoothie with some milk, pour your kefir in there. Just don't cook it. If you cook it, you kill the microbes. And that is something we're currently working on, but potentially could decrease the health benefits. So, yeah.
Wow. So like the goal, like kind of the goal of this research will just kind of, that was just for my own, the goal is basically so you've moved on to human trials, and then what are you hoping like to get out of like the end of this?
In an ideal situation, I think this research kind of started because over the last like 10 years or so, there's been a huge increase in the number, or in the adoption, I guess I should say, of consuming traditional fermented foods, right? So we've always had things like yogurts in the grocery store, and yogurt is traditional, but it seems like every 3 years there's like a new niche fermented food, and it's like, it was kefir, and it still is kefir a little bit. Everybody eats sauerkraut now, or kimchi is a big one, right? But a lot of the times what happens is the microbes that occur in these traditional fermentation versions of them are not the same microbes that are present in the industrialized high production volume versions that we get in the grocery stores. That's not always the case, but we wanted to see if that mattered when it came to a lot of the reason. I mean, they're delicious, but a lot of the reason people start consuming these things is for health benefits. And that's not to say that these fermented foods are useless. Honestly, if you're consuming fermented foods with live microbes in any way, shape, or form, it's likely to have like positive impact on you. But if you're interested in consuming a specific traditional food that has purported health benefits tied to specifically the traditional examples, so kefir, grain fermentation, and you're not consuming one that uses those same organisms, you may not be able to get those same benefits, right? And this can be the same for any fermentation that uses starter cultures. So the goal was kind of to understand how differences in microbial populations or communities influence health benefits of kefir specifically in this case, but sort of by proxy other traditional fermentations potentially. And yeah, we moved on to human trials. We're currently about to start recruiting for a large one at the University of Alberta. My collaborators, Ben Willing and Caroline Richard, have funding from the Weston Family Foundation actually to conduct a large human trial. And we're going to be recruiting for that soon. And the hope there is to expand, because that first trial was really a small group of individuals. We were only able to recruit male participants, which is never ideal because females and males, particularly when it comes to lipid metabolism, have very different metabolisms and outcomes in these things. So this new trial will include more people, a better representation of the different sexes that are, and their responses to these treatments, right? So it'll be more representative of an actual population. And the hope is that the results continue to be good. This will potentially influence things like policy on like fermented foods. We're interested in maybe including like a fermented food group to like the Canada Food Guide, like a recommendation on fermented food consumption would be a cool thing to get out of this. But really just informing people about making more thoughtful choices about what you're consuming, and ideally improving production practices for industrial versions of these foods. Like I said, commercial kefir that you get at the grocery store is not at all bad for you, and it did actually have some benefits when compared to just consuming milk in our trials. It just didn't do as much or as good a job as the traditional culture containing kefir. So that's kind of the outcome. And then for me also, because I'm interested in the ecology and the interactions, I want to understand what specific microbes are interacting and in what specific ways that results in these health benefits. So at MacEwan, I'm working with undergrads to do interaction experiments where we actually, instead of a community of 9 microbes, we take just 2 individual members out and see what happens when they grow on their own or together. And if things are different when they grow together, things like lactobacilli and the yeast particularly, because we know that they're important for the health benefits. So we're currently looking at what happens when I take a Lenti-Lactobacillus kefiri and grow it with Clavaromyces marxianus. And do either the organisms grow better when they're together than when they're on their own in milk? Or do they grow worse? Because interactions can be both beneficial or antagonistic, right? They can fight each other as well. And so by understanding this, we can then maybe narrow it down to specific individual organisms and specific metabolites that are present in the kefir that provide these health benefits. So that would also be really cool. And then I also get the really cool opportunity to look at things like individual interactions and how gene expression changes and how the behavior changes basically in the milk. And then from those individual interactions, we can build up to more complex communities.
Oh, no, that's so cool. And I like it, it seems really complex when you're talking about it with all like the different things. But when you break it down and just be like, we just want to see how two separate ones interact together.
Yeah.
Oh, honestly, I do want to kind of, so you're looking at kefir and the health benefits, but you are also working on spontaneous fermentation in beer.
Yes. Yeah.
As well. So I wanted to ask a little bit about that. And firstly, what is spontaneous fermentation?
So spontaneously fermented beer is originally how beer was made. When we first started making beer as humans, we did not have the ability to isolate a single strain of yeast and then reliably put that strain of yeast back into our beer. It honestly most likely occurred when grains that were left outside got wet from the rain and were allowed to soak for a while, which then resulted in whatever organisms were on them fermenting that liquid and making it alcoholic. So in a spontaneous beer, it differs in that instead of a single strain of yeast being present, you have lots of different yeasts and bacteria present. And maybe it might be better if I actually back up and start with what is conventional beer, because everybody knows beer, but not a lot of people know a ton about exactly how it's made. So for a conventional beer, you take your barley usually, and you mill it. So you crush the grains of barley into not powder, but small pieces. And then you soak that barley in warm water. And what that does is it allows enzymes that are in the barley to convert these big sugars into smaller sugars that the yeast can eat. You then take this wort, it's called at that point. You boil it, you add your hops, you add whatever else you want to add, you cool it down, and then you add your one single strain of yeast to it, and you let it ferment until it eats all the sugars and you've got your alcohol production and your esters and other like acids and things that provide some of the other flavors to beer. A lot of the fruity flavors in beer are from esters. I don't know if you're familiar with like Hefeweizen or like German wheat beer.
A little bit.
Yeah. So it's really famous for having like banana, like bubblegum almost aromas to it. And that's because of a specific ester that the yeast that they use for that beer creates in abundance when compared to other yeasts. So that's what gives it a lot of these kind of specific flavors that certain styles are known for are actually like yeast fermentation byproducts. So that's a conventional beer. You add one yeast, you ferment it for maybe 2 weeks, and then it's ready to package and ship out. Spontaneous beer is different in that it's a very like old traditional process. It's still done a lot in Belgium. And what they do is they still take barley. They also have a large amount of wheat. They still mill it, and they still do this conversion of sugars and other proteins and things down to simpler versions, but they don't convert it as much as you would for conventional beer. Conventional beer, you want as much simple sugar that can be easily fermented quickly. Spontaneous beer, you get more complex because it's a long fermentation. You then do the same thing. You boil it, you add your hops, and then instead of going straight into a tank and cooling down and then having like one strain of yeast added to it, what they do is they put it in a cool ship is what it's called. And it's just like a big beer swimming pool, basically. And it is in this big beer swimming pool, and it's allowed to cool overnight while being exposed to the air outside of the brewery. This allows microbes in the air, so bacteria and yeasts, to kind of settle and fall into the wort. And whatever falls into the wort is then there and able to ferment it if it's able to. After it cools down overnight, the wort is then moved into, instead of like a big stainless steel fermenter like you would see if you've ever gone on a brewery tour, it's moved into oak barrels that have previously been used to make wine, usually. And these barrels are then what this fermentation occurs in. So the bacteria and yeast that are there in the cool ship move into the barrels. Sometimes the barrels themselves have yeasts or bacteria associated with them too. And whatever's there is there. And that community of microbes kind of battles it out to see who's going to become dominant and who can ferment this sugar, who can ferment that sugar, who can break this protein down. And through a series of kind of antagonistic and symbiotic interactions, so symbiotics mutually beneficial for the two of them, right? They create this like very complex, slightly sour beer over the course of a 1 to 4 year fermentation. So instead of 2 weeks, we're talking years.
Oh, wow.
Yeah. So that's spontaneously fermented beer. A really long-winded response, but I'm notorious for that. So
no. So when like in your research, like researching this, what are you, I guess like what is the basis of the research with the spontaneous fermentation? What are you looking at?
So like I said, this is a beer style that's still made in Belgium at a few traditional places. It's called lambic there. It's similar to champagne. Only beer made that way in that area of Belgium can be called lambic. But I work with a partner here that makes spontaneous beer in the same style. Part of it was just, can they do it here? Is it going to work? Is the geography important for it or not? The other part is there's a fair bit known about generally what microbes are present. And at the beginning, we know there's enterobacteria, and then they go away and there's lactic acid bacteria and Saccharomyces yeast, so your typical bread or beer yeast. And then the Saccharomyces kind of goes away, and then long-term there's something called Brettanomyces, which is another type of yeast that is really important for these beers. And it and the lactic acid bacteria tend to kind of take over in the long, like the older fermentations, the 1, 2, 3, 4-year-old fermentations. But while there's been studies on that in Belgium, they typically look at one or a few barrels from the same batch of beer, and they look mostly at just what's there, maybe generally what metabolites are made. So things like lactic acid, acetic acid, your alcohols and your esters like I was talking about. But what we wanted to do was a much deeper dive on these beers because there is quite a bit of variability from batch to batch and barrel to barrel. And we wanted to investigate with this brewery why this occurs. They want to know because if they understand their process better, they can potentially change things about it to make tastier, more consistent beer. We want to know because this is a really cool community for me because similar to kefir, it's got bacteria and yeasts. But unlike kefir, which is really highly adapted, like these communities have been evolving together for thousands of years. They know each other. They are there to work together. They're like relatively streamlined in terms of this is what's needed to get the fermentation done. And while there are slight changes over time in the fermentations are 24 hours. In spontaneous beer, anything can be there. Mm-hmm. But somehow we still end up with this like, well, variable, quite still similar product in terms of it doesn't all turn into vinegar or like terrible, or it always ends up, doesn't always, but mostly ends up being pretty good and having that specific like spontaneous beer quality. If you're a spontaneous beer drinker and you've had enough of them, you can pick it out. Yeah. So it really is a cool opportunity for me to study a highly variable community of microbes that is really quite drastically changing over time and has a ton of potential interactions in there to make an extremely complex product in terms of the number of metabolites that are present. When we talk about metabolites, we often talk about like a few hundred or something like that. Kefir, with some of the chemistry people that I collaborate with, we've gotten close to like 700 different metabolites identified. In beer, we're well over 1,000 in these spontaneous beers. So there's a ton of metabolic diversity there. And so it's just a really, really cool community. There's so much going on. it's such a long time period, so it allows for lots and lots of changes to happen, which for me is really interesting. It's a little bit hard to study when you're talking samples over years, but it's really interesting, and it's really cool to see, well, does beer brewed in the fall versus in the spring look the same or different from each other? Do different batches look different from each other, even if they're brewed within a week of each other? How much does the barrel matter for this? What happens when we have slightly different starting compositions? Does that lead to the beer at 2 years looking very different, or does it actually not matter too much what happens at the beginning? Like, all of these are kind of somewhat unanswered questions for both the beer industry and also for myself, like I said, as someone that's just interested in, well, what are they doing? Are they getting along? Are they fighting? Are they, like, how does that influence who's there later on and what the beer actually like tastes like or smells like? So that's kind of like the basis of it. There's also, sorry, I'm continuing, but there's also the flip side of what's called mixed fermentation beer. And that is where, similar to conventional beer, where you make your wort, you put it in a tank, you pitch your single strain of yeast. Mixed fermentation beer, you make your wort, you put it in a fermentation vessel. It can be barrels or it could be a tank, and you pitch a mixture of organisms. So you would pitch some lactic acid bacteria, some Brettanomyces, some Saccharomyces, maybe something else. So the flip side of this is that by learning about these spontaneous beers and maybe what makes them tick, so to speak. So, barrels that have more like fruit characteristics, do they have different organisms in them than barrels that have more like leathery is a flavor that you get sometimes. So like leathery characteristics, or ones that are more sour or less sour. Do they have different organisms in them? Is it something that we can maybe replicate and recreate with mixed fermentation as well? So it's kind of two-sided, where we learn more about spontaneous, and that allows us to make better spontaneous beer. But then there's also the potential for making entirely new strategies and production methods for producing mixed fermentation beers with a broader range of flavors, better consistency, just better quality. So that's kind of like the double-sided nature of the fermentation, beer fermentation research. Yeah.
Well, so you did mention that you are working with a brewery here in the city. From working with them, have you been able to discover anything as of yet? I know this research is still fairly new, and like you said, it takes years instead of weeks.
Yeah. So
to do it.
We are working with Blind Enthusiasm Brewing. They're the only brewery in Canada and potentially North America that's purpose-built to make spontaneous beer. The brewmaster there actually trained at Cantillon in Belgium, which is like the mecca for this kind of beer. If you're into it, like, everyone knows Cantillon if you talk about it, if in the right circles. So it's a really great opportunity for me to study, again, something that I think is really cool and really and have the opportunity to work with quite literally like some of the best people in the world at making this kind of beer. And to have it in Edmonton's really lucky for me, and to have a partner that's willing to put the money and time investment and is willing and open to change processes based on scientific data, because a lot of the times maybe they know it works and they're happy that it works, and that's totally fine, because you can make fantastic beer as more art than science, but they really kind of want to blend the two together, and that's really cool for me to be able to work with them on that. We have done some things with them where they've changed certain things about their processes in-house. We've also done some work looking at differences in barrels. Like I said, past research in Belgium tends to look at a small number of barrels. We are currently looking at well over 100 barrels over a 2-year time period at the brewery, and we've identified like some variability in the communities that are present in the barrels over time, and are currently looking at how that variability impacts like flavor outcomes within those barrels, whether that's metabolites or potentially in the future actual sensory analysis. So that's where we're at right now. Like I said, it's fairly early. We're also doing something similar to what I talked about in the kefir, where we are isolating organisms out of these beers to then put back together in meaningful and specific ways. You can't perfectly replicate a truly spontaneous fermentation. It's not like kefir where you have this still have a starter culture, but it's the grain starter culture, and you put it in. In spontaneous beer, it is truly dozens of organisms start out, and then it's random every single time, or like we like to call it like highly stochastic, and like my field is the term that we use for it. And so you can't fully recreate that with the reconstituted community, but it does allow us to look at things like, well, what happens when this yeast that seems to show up sometimes but not all the time is there? Does that actually matter for flavor? And so in this kind of next phase of the project, where we're looking at continuing the spontaneous, but also getting a bit more into making our own communities, manipulating them, seeing what happens, we're hoping to identify maybe new yeasts that we can add to beer to make like unique flavor profiles. I'm working with, again, Ben Willing at the U of A, Wendy Wismer at the U of A whole team of microbial ecologists, food fermentation chemists, and sensory scientists. And we're actually this fall getting some 800-liter pilot scale fermentation tanks that we're going to be able to do experimental beers in. And then we're actually going to be able to ferment, package, and analyze these beers. How does the gene expression change when we do things like change the oxygen level in the beer or add certain organisms? How do the metabolites they make change? And then we're actually going to be able to do sensory panels where we get individuals that are trained on tasting these beers to blindly taste the different experimental fermentations and see if we can then generate identifiable differences in these beers. Because the one downside of metabolomics, or the analysis of like broad numbers of metabolites in these products, is that, well, we can identify flavor active compounds and we can kind of say this compound tends to be associated with this flavor, like isoamyl acetate is an ester that gives you banana, right? Or phenylethyl alcohol and phenylethyl acetate tend to give you like floral rose-like aromas. They're actually what's used in like rose perfumes and flavorings. And we can potentially tweak fermentations to make more or less of these compounds depending on what the brewer is looking for. But these metabolites interact in a very complex way when they're all together. And like I said, you have over 1,000 in these beers that we can even identify. There's more than that even. And we don't actually have a way of modeling like this level of this with this results in this specific flavor. Individuals and the human like aroma and like tasting sensory experience is not currently able to be modeled by instruments or like data analysis. So we can say, well, this is likely to result in this, but without sensory, we can't actually say for sure that there is a definite difference. So being able to add that sensory component then kind of takes us to that final step of, we change this thing, the organisms behave differently, they make different metabolites, and people can actually taste the difference when they drink the beer, which is really cool for me because it's, you know, I get to see how tweaking environments or communities changes behavior and outcomes. And then for the brewery, they get to see this is actually like an identifiable, discernible difference. And it's beneficial for them because it could be that people really like it and maybe they want to try doing it. Or they get the opportunity to try this on not a full production scale and see this is really horrible and we don't like it, because that is something that happens when you're doing experimental fermentations, right?
Yeah.
But it's really exciting. It's still, like you said, it's relatively early days on this, partially because the spontaneous stuff takes so long to analyze. But we are hoping to start getting some of the information out there in the next year or so. Ideally, some of this would also help more than just the industry partner. People in the brewing industry that want to make better mixed fermentation beers in general could potentially use some of this information as well. Yeah.
Wow. No, that's really cool. And it'll be exciting to see where that ends up once you're able to get to that spot. I know we've talked about like beer and the health benefits of kefir and all of that. And just to kind of like wrap up a little bit, I was wondering like what in your eyes makes this research timely? Like, why is it important and relevant today?
It feels like every few years there's a new fermented food, superfood, right? That's coming out, and people are aware of and care about live microbes and fermented foods and the potential health benefits. But there's a lot of misinformation out there. I can even say that I have found references to some of my own papers on health food blogs that I'm just reading. I'm like, that's not at all what that paper says, or this is not actually true. Things like I said earlier, well, in mice, kefir seems to improve this specific type of cancer, and then now you have people saying, drink kefir if you have cancer. If you have certain types of cancer, the microbes in kefir could actually cause infections and be more damaging for you. So this should be maybe my thing of, if you're really sick, please talk to your doctor before you consume like live microbial foods.
Yeah, please consult with the doctor.
Yeah, exactly. You would you'd be amazed at the amount of emails that I get from people asking if their like family member with cancer should be drinking kefir or this or that. I also just think like probiotics again is a big one, and probiotics and fermented foods are kind of intrinsically linked with each other because a lot of the organisms we think of as probiotics have come from fermented foods. But this also comes back to my kefir research, where if you look at, there's the International Society for the Advancement of Pre and Probiotics. They have these expert consensus statements that they've put out over the last decade. And the one for probiotics states that like the requirements for a probiotic are actually very, very narrow. The European Union has yet to approve a single like health claim for probiotics actually. And a lot of these benefits that are reserved are not just species specific. So I was talking about Lentulactobacillus kefiri. That's a kefir specific bacteria, in case you could not tell from the name. There are within that species going to be potentially dozens of different strains that are all the same species but slightly different from each other, right? We're all humans, we're the same species, but I don't look the same as you, or everybody has differences. In microbes, it's not necessarily in how the cells look. It may be that one of them produces a specific unique metabolite, or can consume a specific sugar or something like that that's slightly different. And that strain level difference can actually be the difference between seeing those health benefits or not. And so this is why it's so important that proper research is done on what specific microbes are actually responsible and necessary for any health benefits, and that we actually do properly controlled human clinical trials before we start to make claims on these functional fermented foods. So that's on the kefir side why it's relevant. I think it's a really important thing. Again, please, outside of the talk to your doctor example, don't stop eating fermented foods. They're still all going to be really good for your gastrointestinal health and your gut microbiota and those things. But again, if you're looking for very specific health benefits and you want to make health claims around products, you need to know that you've got the right stuff there, right? Yeah. The beer side, I mean, beer is always kind of relevant. Craft beer has been growing quite a bit. Mm-hmm. There's a big push in the beer industry and in Canada, especially for increasing these types of industries. They can employ quite a few people. And spontaneous beer in particular has been getting more and more popular over the last few years. I would say not everybody likes beer. Lots of people like beer. It's a really easy one. Whenever I say that I do beer research, people always want to talk about that. So that one is interesting and I think pertinent in terms of just, again, people are always looking for the next cool thing. If we can potentially find a new way to make mixed fermentation beer, or a way to make new flavor profiles within the beer, I always say like there's no such thing as the perfect beer, because everybody's tastes and palates are going to be different. Some people, the perfect beer is Bud Light. Some people, the perfect beer is like really, really aggressive, like bourbon barrel aged stout, or like a smoked like Rauch beer, or lambic beer like Cantillon, or spontaneous fermentation. The more different ways that we can purposefully broaden the types of flavor profiles that we have for beer, the more likely someone is to be able to find their perfect beer, or for a brewer to be able to make their perfect beer, right? And being able to do stuff that nobody else is doing is really beneficial when you're in a very competitive industry, right? So that's kind of it. And then again, understanding microbial interactions often has farther reaching, not consequences, but implications than the specific community that you're studying. So yeah, we might find something in beer, but it could be that it's also a type of interaction that occurs in soil communities that are important for agriculture, that kind of thing. So there's potential to then collaborate with soil microbial ecologists, because that is a thing. There are people that work on soil microbial communities and specifically how agricultural conditions influence them, and they influence agriculture. So yeah.
Well, I want to thank you so much for being here and taking the time to talk with us today.
Yeah, it was tons of fun. Like I said, I always like talking about beer, so.
Thank you. Well, that's all we have for today's episode of Research Recasted. If you want to support this podcast, you can visit Research Recasted on your favorite podcast platform to find new episodes every 2 weeks. Also, don't forget to check us out on Instagram at Research Recasted, where you can give us a like, leave us a follow, or send us any follow-up questions you have from today's episode. This has been Research Recasted, the Knowledge Mobilization Podcast, brought to you by the Office of Research Services and the Faculty of Fine Arts and Communications at MacEwan University.